Okay, we can start. And okay, I hope that my presentation could be useful for your work, for your activities, and at least for your background, because it's concern basic research. And okay, so. Okay, the title of my talk is High Throughput Transcriptomic and Metabolomic, Grape Quality, Climate and Grape Cultivars. So many things in this title. I tried to divide my presentation in three major points. So a brief introduction of the principal omic approaches, the focus on transcriptomic and metabolomics in grape, the platform and the technological advances, and the final part of the presentation will be focused on some of the most recent results obtained by these two important analytical approaches in grapevine. So in order to talk about omic approaches, we have to briefly remember the flow of biological information of a cell. So the genome contains all the information of cell life. Genome is the total DNA of a cell, and inside of the genome we have several genes that are sequence of DNA that contain information for the synthesis of protein. One gene must be expressed in order to produce a protein. So in the cell we found the presence of a molecule named transcript, that is a molecule of RNA that is produced by the sequence through the sequence of a gene. Thanks to this molecule, we have the production of a protein, a specific protein, and the activity of proteins can produce a metabolite. In a particular moment of cell life, we have the expression of several genes, not all, but several genes. So we have the presence of many transcripts into a cell, as a consequence, the production of many proteins, and as a consequence, the production of many metabolites. The final characteristic, the quality trait of our samples, if we are talking about grapes today, are given by the total amount of metabolites and the activity of proteins. Each step of this important flow can be analyzed by omic approaches that adopt an holistic view of the molecules that make up a cell tissues of organ. In particular, genomics is the study of the total genes contained into a genome. The transcriptomics is the study of the total transcript, proteomics, the total proteins, and finally metabolomics of the total metabolites. Concerning transcriptomics, okay, the transcriptome is the total amount of transcript and depend on gene expression. Therefore, the transcriptome can change qualitative and quantitatively according to cell type, developmental stage, or in response to external condition or physiological state. There are two approaches, two principles that can be used for the determination of the total amount of transcript. There is the digital analysis of sequence, that is simply the count of the number of times that a particular sequence is present into a sample. In that case, we can count the transcript referred to each genes, and we can determine the presence of this transcript and determine the expression of each gene. Or we can determine this important information through an indirect approach that is based on an hybridization step in which the total transcript of a cell will react with a small support that could contain many sequences that represent almost all the genes of the genome, and this reaction produces fluorescence signals that are proportional to the level of expression of each gene. In grapevine, we have a lot of works performed on these two by transcriptomics. We think that the first one could be considered the work performed by Davis and Robinson in the 2000, in which the authors tried to analyze genes expressed during ripening. Then the history of the transcriptomic of in grapevine is defined by the technological advances that improve a lot the powerful of these approaches. In particular, in the 2007, there was the most important step in the improvement of this transcriptomic experiment and platforms that consist in the genome sequences of grapevine. So thanks to the work performed by the Italian-French consortium, in the 2007 we were able to find that 90,971 genes are present into the genome of grapevine. So now we know exactly the sequence of all these genes. This important information allowed to improve the transcriptomic experiment. In particular, now we are the possibility to have small support, name arrays, macroarrays, that are able to represent more than 90,000 genes. So in one single reaction, we can monitor the expression all these genes. And also, the digital count of transcript was improved by this knowledge, and together with the recent technological advances in the next generation sequences method, now to the grapevine we can apply the method that is called RNA-seq approach that gives a more complete information about gene expression analysis. Concerning metabolomics can generally be defined as the study of complete set of metabolites. Like the transcriptome, the metabolome is dynamic, so is not always the same, varies by cell type, developmental stage, and the response to different stimulus. But unlike the transcriptome that is composed by transcripts that are similar at chemical level, the metabolites are different, so is not present. We cannot use a single analytical platform to cover the chemical diversity of all metabolites into our sample. The mass spectroscopy is the most commonly used method to investigate, identify metabolites that is often associated to the liquid chromatography for the determination of non-volatile metabolites or gas chromatography for the volatile metabolites. Both these powerful analytical approaches produce a very huge amount of data, and a common step for the analysis of this data and to try information from this data is made by bioinformatic tools. So it's a necessary step for the interpretation of data obtained by these approaches. So at the end of this analysis, we can determine genes and metabolites that significantly modify their expression or accumulation in different samples that we can analyze. In fact, at the end of this analysis, we can profile the gene expression of all our genes that we can monitor, or all metabolites, and we are able to find the decreasing or increasing expression of these molecules. What are the principal developmental and physiological processes analyzed by these approaches in grapevine? Of course, the berry formation and ripening, so the entire developmental process of berry that could be divided in two distinct parts, the berry formation before veraison and berry ripening after veraison. And also the responses of plant or of berries to biotic and abiotic stresses. In particular, if we will focus on berry formation and ripening, I would like to show you an important study performed in the 2012 in our lab, based on the hybridization approach, so transcriptomics, that allow us to determine the expression of 29,000 genes. We analyzed the gene expression in 52 samples that represent different organs of plant in different developmental stages. And we can focus on the berry formation and ripening. We analyzed 5 developmental stages of berries, 2 before and 2 after veraison. And one important result that we obtained by this analysis is the determination of many genes that are a characteristic expression that can exactly separate the two phases of berry development. So we found genes that are high expressed during the vegetative phase of berries, so the berry formation, and then their expression sharply decrease during their maturation. And on the contrary, we found many genes that are expressed at very low levels during the formation of berry, and then rapidly increase during the berry ripening. These genes are genes related to sugar accumulation, softening of the fruit, and also secondary metabolism that contain genes involved in aroma or organoleptic properties of the fruit. Another important work that we perform in our lab concern the next question. So we now know the genes that are expressed during ripening, but how the expression of these genes could be affected by the growing condition. So we analyzed a single clone of these varieties, that is Corvina varieties, typical of Verona province. And this clone of Corvina was grown in 3 different zones of Verona province during 3 vintages. We found that the vintage has a very strong effect on gene expression, more than the growing condition. But we are also able to find genes that could clearly separate the 3 growing conditions. So vintage, but also genes which expression could distinguish the 3 growing condition of this variety, and also genes that can able to separate the growing system of this cultivar. Another work performed recently in our lab was about the white variety Garganega, another local variety of Verona province, and again the question was how the expression of genes involved in ripening of these varieties can be affected by, in this case, by differences in the soils, in the growing condition, yes, but also in the soil. So also in that case, we applied transcriptomic and metabolomic approaches. We found that this cultivar can react to the different growing condition in terms of metabolomic and transcriptomic changes, and also that genes and metabolite that belong to the phenylpropanoid metabolism show the strongest responses to these differences. Another important study, again concerning the berry formation ripening, was performed on 10 Italian varieties, 5 red and 5 white, that represent different regions of Italy. In this study, all these varieties were grown on the same experimental field, so in the same condition, and we collect 4 developmental stages for each, with the aim to, in that case, found genes that commonly modulate their expression. So in the other study, we found genes that modulate their expression in different conditions. In that case, we work with different genotypes, but we found common genes that are involved in the berry formation and ripening. So it's another important step in the knowledge about this important process at molecular level. Concerning the responses of grapevine to biotic and abiotic stresses, I have the time to show you only a few examples, but in the literature there are a very huge amount of works in which authors try to find differences in gene expression between, for example, susceptible and resistant cultivars to many pathogens. I represent here the most important, but there are a lot of examples of these studies. And also there are a lot of examples of studies performed by transcriptomic and metabolomic approaches that answer to the question how the plant can react to the water limitation or drought. So we found these are 3 of the most recent work, but there are so many. And also in that case, authors are able to find that grapevine berries respond to drought by modulating several secondary metabolic pathways in terms of gene expression and also in accumulation of metabolites. And these important metabolites, this result is very important because these metabolites are directly related to the sensory features of the mature fruit. There are a lot of application also to the response to the berry, to the plant and the berries, in particular to the temperature, and also to viticultural practices. We perform in our lab with a collaboration with several other universities in Verona, works aimed to analyze genes which expression was affected by viticultural practices such as cluster thinning or defoliation at different developmental stages of berries. I would like to finish my presentation by showing you our last work performed again by transcriptomic and metabolomic approaches that was published at the end of the last year. And this is an important work for my opinion because we, in this work, we analyze differences in response among different genotypes. We analyze 6 red grapevine genotypes. And the condition in which we analyzed these genotypes was the post-harvest dehydration. That is a peculiar practice of Verona province for the production of special wines, such as, I don't know if you know, but Recioto and Amarone wines that are very important for our region. And the cultivar that is mainly employed in this production is Corvina, that follow a particular dehydration process, and we compare the reaction of the response of other genotypes to this important process in order to identify molecular and metabolomic changes that characterize this process and the difference in the different genotypes. Briefly, this process, when the berries are fully reach the full maturity, were detached from the plant and stored in drying rooms with controller temperature and humidity conditions. During this process, there is an increase in solute concentration due to the evaporation of water, the physical and chemical changes, and also the modulation of primary and secondary metabolism. All these changes could be due to many factors, such as the berries that enter into a so-called ripening senescent phase, that is a follow phase after the maturation, or the berry or berries can respond to different stresses, such as the detachment from the mother plant, the dehydration stress due to the water loss, and also other abiotic factors or the infection of botrytis. By this work, you can see here the different kinetics that characterize the 6 genotypes that we analyzed. We are able to identify common molecular responses, so genes that in all these 6 genotypes, independently from the genotype and the kinetics in water loss, are commonly upregulated during the process. This is an important information because these results demonstrate that berries are not, and the cells are not died, but they are still alive and can react actively to this particular process. And also we found genes that are commonly down, genes that are commonly switch off, so their action, their activity is not important during this process. But simultaneously, contemporaneously, we found also genes and metabolite accumulation that could characterize the different responses of the 6 genotypes analyzed. In particular, we focus on the stillbed metabolism. In this part of the graph, we can see the accumulation of resveratrols and of its polymeric forms in the 6 genotypes during the time. And in this part of the slide, we can see the expression of genes that are involved in the synthesis and the polymerization of resveratrol. And the expression of genes exactly mirror the metabolites accumulation. And so these important results show that these 6 genotypes can clearly distinguished by this important metabolism. So, my conclusions, the transcriptomic and metabolomic analysis allows to obtain a large amount of information regarding transcript and the metabolite changes that characterize a particular developmental process or a specific stress responses. The technological advances are constantly improving the analytical power of omic approaches, and these approaches have a more applicable to the real condition in the field, and could try to answer to the real question of the grower and to the viticultures. But there are some limits, of course, the high experimental cost, in particular for the transcriptomics, because the technological advances improve a lot the platform, and the costs are cheaper than some here before, but still high, unfortunately. The needs of informatics support, because the data are very a large amount, and so it's difficult to manage this data. And finally, the limited information about a single gene or single metabolite function. So through the metabolomic and transcriptomic, we obtain a general overview. We can focus on a single genes, but to have the final demonstration that this genes is involved in this process, we have to take the genes and perform functional analysis in the other kind of experiment. These are all the people that are involved in most of the work that I show you, in particular my colleague from the University of Verona, the Professor Mario Pezzotti, my colleague Giambattista, Silvia Da Santo, and Marianna Fasoli, that are involved in these studies. And thank you for your attention.